Hello everybody, welcome to the T.Y.B.Sc class of Enzymology, that is paper 3 MB333. We are going to start this course with the topic 1, namely enzymes. The topic 1, unit 1 contains structure of enzymes and with special reference to the methods that are used to determine the amino acid residues at the active site. These methods can be categorized into two types, physical methods and chemical methods. The learning objectives of this particular lecture can be defined as by the end of the lecture, the student should be able to describe the methods of active site mapping which deal with the enzyme modifications by chemical procedures, by proteases, and by site-directed mutagenesis. The methods can be summarized as the chemical methods, which include trapping of the enzyme substrate complex, use of substrate analogs, and enzyme modifications by chemical procedures, enzyme modifications by treatment with proteases, and enzyme modifications by site-directed mutagenesis, of which the trapping of the enzyme substrate complex and use of substrate analogs we have already studied in the earlier lecture. In today's lecture, we shall study the rest of the three chemical procedures, namely enzyme modifications with chemical procedures, enzyme modification by treatment with proteases, and enzyme modification by site-directed mutagenesis. The physical methods, namely X-ray crystallography and nuclear magnetic resonance, will be dealt in the further lectures. So coming down to the study of enzyme modification by chemical procedures, we know that at the active site of the enzyme, the amino acids that are present are showing their R groups which are free to bind to the substrate. Enzyme being a protein made up of amino acids, and the specific amino acids that are present at the active site whose R groups are exposed or free to bind to the substrate are playing a very important role in the substrate binding and further catalysis of the substrate or conversion of the substrate to the product. Keeping in mind this particular activity of the R groups, to study the amino acids that are present at the R groups, we have exploited this particular characteristic of the active site or of the R groups of the amino acids at the active site by blocking or inactivating the R group by using some chemical. If the R group of the amino acids that are present at the active site of the enzyme is inactivated by reaction with some chemical, obviously there is no scope for this particular R group now to bind to the substrate and convert the substrate to the product, and thus the active enzyme is now converted to an inactive enzyme. And so loss in catalytic activity of the enzyme due to the treatment with a particular chemical which inactivates the enzyme can be referred to saying that this particular amino acid whose R group is modified may be present at the active site of the enzyme. But there is one little bit of limitation to this particular method, and that is the loss in catalytic activity of the enzyme may also be due to modification of the enzyme, which is not actually present at the active site, but which is responsible for the disturbance or the change in the tertiary structure. As we have seen earlier, in the levels of the primary, secondary, and tertiary structure, the tertiary structure is kept stable or kept together due to the bondings between the R groups of the amino acids that are confronting each other in either the secondary or the tertiary structure. And so if these R groups are affected by the chemical, it is very much possible that either the secondary or the tertiary structure of the amino acid will be affected, and thereby the exposure of the active site or the conformation of the active site may be affected. So it becomes very much necessary to distinguish between these two situations. That is, whether the inactivation of the enzyme is due to the inactivation of the R group that is present at the amino acid that is present at the active site, or the inactivation of the enzyme is due to some change in the tertiary structure which has distorted, which has disturbed the structure of the amino acid, which now cannot bind to the substrate. This can be done by a very simple experiment, that is by carrying out the same experiment but in presence of the substrate at saturating concentrations and in absence of the substrate, like we have done it earlier. The difference in these two types of experiments that we are doing are, in absence of the substrate, the R groups of the amino acids that are present at the active site are available for modification by the chemical and thus will get modified. But when you are treating the enzyme prior to the chemical treatment or before the chemical treatment with saturating concentrations of the substrate, it is now likely or it is now obvious that all the R groups of the amino acids that are present at the active site have been bound to the substrate. After this, when we are treating it with the chemical, the R groups of the amino acids at the active site are not available for the activity of the chemical, and so they are not inactivated. Whereas the R groups of the amino acid which are present elsewhere in the enzyme are inactivated. Thus, at saturating concentrations of the substrate will bind to all the substrate binding sites of the enzyme, and it will prevent the amino acids of the active site from being modified or from being inactivated. So, to conclude the results of this experiment, if the enzyme loses activity in absence of the substrate, but it remains active in presence of the saturating amount of the substrates, we can definitely say that this particular amino acid is present at the active site of the enzyme, which can be illustrated in this way. So you have the R group at the active site of the enzyme. You are saturating it with the substrate. So the R group now binds with the substrate, and now we are giving or treating it with the particular chemical. So in presence of the substrate, the chemical treatment cannot modify the R group because it is already bound to the substrate. When you isolate this enzyme substrate complex and study its activity, we find that the enzyme is in the active state and it has converted the substrate to the product which has been released. So modification of the catalytic site will result in complete loss of activity. By using such type of experiments, we have found that the amino acids like histidine, cysteine, serine, methionine, tyrosine, aspartate, glutamate, lysine, and tryptophan are the amino acids which are present at the active site of many enzymes. To exemplify the chemical modification, we now see the first example, and that is use of iodoacetate for modifying the R groups. Iodoacetate specifically binds to the histidine at 119th position or the histidine at the 12th position of the enzyme ribonuclease. So here we can see in this figure, this is histidine which is reacting with iodoacetate. The iodoacetate has transferred this acetyl group to histidine, converting it to carboxymethyl histidine residue. Since this acetyl group now has bound to a place which probably is important in binding to the substrate, there is no scope for the substrate to bind at this particular position. And so the enzyme becomes inactivated. In this way, it was found that the histidine at the 119th position of the enzyme, or the histidine and the histidine, sorry, at the 12th position of the enzyme is present at the active site of ribonuclease. The second example is the use of ethyl malimate. Ethyl malimate acts on all the cysteine residues in the enzyme. So cysteine amino acid is a sulfur containing amino acid which has the SH groups. This N-ethyl malamide, it binds to the SH group of the enzyme, and SH groups are the groups which are very important in binding to the substrate, and thus the ethyl malamide, it binds and it blocks the substrate binding site of the enzyme, and thus it modifies the enzyme and inactivates the enzyme. In this way, it was found that if the enzyme is inactivated by the use of N-ethyl malamate, then definitely cysteine residues are present at the active site of that particular enzyme. The third example is by inactivating the enzyme, the amino acids, with the help of photooxidation. Oxidation by oxygen in presence of a photosensitizer such as methylene blue or rose bengal inactivates the enzymes, because it inactivates certain amino acids by the process of oxidizing the amino acids. The method is non-specific, and so it may affect all the types of amino acids, and thus it has limited use. But the method can be made a little bit specific, where you can choose the photooxidation, or you can choose a photosensitizer which under certain condition can oxidize only a single amino acid or a single type of amino acid in the protein, and then we can come to some particular conclusion. So to exemplify this, it was Wested in 1965 who mapped enolase in presence of rose bengal, or photooxidized enolase in presence of rose bengal, which affected only a single histidine residue at a particular position. And this histidine was inactivated by photooxidation in presence of rose bengal, and so we could definitely say that this histidine is present at the active site of enolase. To confirm this, the same experiment was carried out in presence of magnesium ions, and it was found that magnesium ions which bind to the histidine and protect it from photooxidation. So this confirmed that it is the same histidine residue which is present at the active site of enolase. These are some few more examples of the chemicals, or of the determination or finding out the amino acids at the active site using certain chemicals. So it is papain. In papain, which was found that cysteine is present at the 25th position of the active site, and this was found by use of iodoacetamide and parachloromercury benzoate. Then lysine was present at the 41 position of ribonuclease, which was detected by the use of dinitrofluorobenzene. Tryptophan was present at the active site of lysozyme, which was found with the help of photooxidation. Ethyl malamide and iodoacetate we have already dealt with or discussed earlier. Now coming down to the next method, that is enzyme modification with proteases. The modification of modification of enzymes by proteolytic enzymes can definitely give us an information of the active site, because proteases basically are responsible for hydrolysis of the enzymes or hydrolysis of the proteins. And enzymes being proteins, the proteolysis of the enzymes is going to disturb the structure of the enzymes. And if this enzyme is inactivated by the activity of proteolytic enzymes, and if we can further focus this method to find out which amino acids were inactivated, this will definitely give us information regarding which amino acids are present at the active site of the enzyme. So to exemplify it with, we first select the example of aldolase. So when aldolase was treated with proteases, which was removing the amino acids from the C-terminal residues, the loss of tyrosine from the C-terminal of that particular protein or that particular enzyme reduced the activity of aldolase. And this definitely suggested that tyrosine is present at the active site of the enzyme. The second example is ribonuclease. To cleave or to hydrolyze ribonuclease, the enzyme cetylicin was used. Cetylicin is a protease which is produced by the microorganism Bacillus cetylis. This protease or this enzyme, it splits ribonuclease into two parts or two units. The first unit is called S-peptides, which consists of the first 20 amino acids in the primary structure. And the second unit that is cleaved is called as the S-protein, which consists of the remaining 104 amino acids of the enzyme. Individually, when the protein or when ribonuclease was split into S-peptide and S-protein, and these individual units or these individual fragments were tested for the activity, it was found that none of these proteins had catalytic activity separately. But if possibly they are mixed in equimolar proportion and they join together again, the enzyme activity is restored. So this suggests that S-peptide and S-protein both contribute to the active site of the enzyme. These fractions can further be proteolyzed to locate the exact amino acids which are responsible for the activity of that particular enzyme. The next method is enzyme modification by site-directed mutagenesis. Mutagenesis is the process of bringing about mutation in a particular compound, and site-directed mutagenesis is bringing about the mutation at a particular predetermined site, that is at a particular locus in the gene or at a particular locus in the DNA, which is responsible for changing a particular amino acid in the protein. This we have already studied in our second year in the process of transcription and further translation and how mutations affect the transcription and translation. So, by carrying out site-directed mutagenesis, either by recombinant DNA technology or by use of specific mutagens, we can introduce specific amino acids at particular positions. Or rather, in very specific or in very right words, the amino acids that are present in the name or the previous enzymes can be substituted by the different or newer amino acids. And then it is very important, or it becomes very easy for us to conclude that if the substitution affects the activity, then the earlier amino acid is present at the active site and responsible for the activity of the enzyme. So to exemplify this, we have chosen the example of trypsin, in which Craig and his associates in 1981 did this experiment whereby they replaced aspartate, which was present at the 102nd position, by asparagine. And this replacement resulted in 1000 times loss of the activity of trypsin. And so we can definitely say that it is aspartate which is present at the active site and responsible for the activity of the enzyme. Similarly, in the second experiment, which was done by Raines and his associates in 1986, with triose phosphate isomerase, they replaced glutamate at the 165th position by aspartate. And this also similarly resulted and about 1000 times less activity of the enzyme, by the way of which we can conclude that glutamate at the 165th position is present at the active site and is contributing to the activity of triose phosphate isomerase. So students, I hope and I am very sure that you have understood all the three methods of mapping of the enzyme substrate, mapping of the amino acids that are present at the active site of the enzyme that we have studied today, namely enzyme modification by using chemicals, enzyme modification by using proteases, and enzyme modification by site-directed mutagenesis. Thank you.